Good morning, everyone. I'm Solène Brohard, and I'm part of the CNRGH, the National Center of Research on Human Genomics in the Laboratory of Bioanalysis. I present my poster on the maturation study of organoids from single-cell RNA-seq data analysis. The organoids are three-dimensional structures derived from adult or pluripotent stem cells. They are cultured in vitro to recover the architecture and the functionalities of a target in vivo organ. The concept is based on the self-organization process of cell differentiation. The cell repertoire of the targeted organs is expected in organoids to consider it as mature. The cell sources and the protocol applied depend on the targeted organ. For this preliminary study, we used kidney organoid data. Subramanian et al. Proposes freely available data of single-cell RNA sequencing of kidney organoids cultured from human-induced pluripotent stem cells from two different commercial cell lines and at different times of maturation, day 0, day 7, day 15, and day 29. From these datasets, we applied a bioinformatics workflow based on the R-Storat package. We filtered low-quality cells like doublets or degenerative cells. The single-cell RNA-seq workflow was first launched on each individual data. To assess the maturation of organoids, we decided to add 3 analysis steps. We first integrated datasets at the different maturation days to compare the proportion of cell populations. We also looked at the cell trajectory on the integrated datasets to visualize cell differentiation across the different times of maturation. We finally explored the variability of cell population present in the sample and their distribution in clusters. For the integration part, we used the canonical correlation analysis called CCA. We showed the results in the integration of the Alcium cell line maturation only. We first observed that the CCA had correctly integrated datasets by looking at the UMAP visualization before and after integration. The clusters were defined after CCA. The comparison of proportion of cells in clusters for each sample at each day of maturation highlighted a maturation gradient, an early maturation represented by clusters composed of day 0 and day 7 cells, an intermediate one with clusters composed of cells from whole maturation days, and a late maturation represented by clusters composed only of day 15 and day 29 cells. We also found specific clusters for day 29 corresponding to stromal and proximal cells. We then tested the cell trajectory approach with monocule 3 on the integrated datasets of all stem cell line. We observed the 2 early and late levels of maturation, and we visualized that the clusters specific to day 29 were at the end of the trajectories. We also observed the gradient of organoid maturation from day 0 to day 29. Finally, we wanted to test if we can define the maturation by estimating the variability of cell population and their distribution in clusters. We used the Jensen-Shannon divergence score that compared the proportion of cells in each cluster between 2 samples. We applied these statistics on all integrated datasets from the 2 commercial cell lines. We first observed the little divergence between the 2 cell lines for the same day of maturation, meaning that the culture protocol gives similar organoids at the same age, not depending on the origin of the human-induced pluripotent stem cell line. By comparing days of maturation for the same cell line or across the 2 cell lines, we observed a high divergence between day 0 and day 29, day 0 and day 15, and day 7 and day 29. But we also observed that day 0 and day 7 and day 15 and day 29 showed similar population and proportions of cells. Thanks to this preliminary analysis, we defined 2 distinct early and late maturation levels in kidney organoids. We now plan to integrate different data sets using different protocols, cell lines, and maturation ages to the study to see if we find similar results. We also want to do the same exploration on organoids of another target organ. The final objective is to use machine learning-based approaches to develop a method that can estimate the maturation level of organoids. Thank you for your attention.